Okay, everybody. It is officially just after 8 a.m. my local time, 10 a.m. Eastern time. So we're going to kick it off to today's webinar and workshop on gastrointestinal MPS. As we begin, I have a short disclaimer that I need to read. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subject, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3Rs Collaborative nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So I'm going to talk a little bit about both my organization. We have a representative from the IQ as well as this webinar series. So the 3Rs Collaborative, our MPS initiative's goal is to really collaborate to accelerate the adoption of MPS in scientific research. But our wider mission is just to advance better science for both people and animals. We do collaborate broadly across different industry. This is just a picture of our board of directors and leadership teams and their logos. And our Microphysiological Systems initiative is again about that adoption in coordination with animal studies. We have over 45 member institutions and over 70 members involved. And our initiative really aims to provide thought leadership and expert consensus, discussion, collaboration, and sharing, engagement between developers as well as regulatory agencies, external partnerships and collaborations, as well as just resources to facilitate increased engagement with developers. We've got three main work streams that we accomplish these goals. We interface with end users and regulators, such as today. We publish papers, host meetings, and also have our technology expo. We do engage consistently with end users. This is some of our progress from 2021, 2022, and 2023. We've been a regular presenter at the MPS World Summit. We post the recordings of these on our workshops. Today's presentation will be recorded, although I encourage you to stay till the end so you can participate in the roundtable Q&A, but recordings will be posted on our website along with our other workshops prior. For regulatory work, again, lots of progress here. We have talks at the MPS World Summit. We've got a presentation or a publication in mind that we're drafting. We also work with regulators as well, where we speak at the 3D Tissue Summit and have a movement to establish a PPP. We also provide education and thought leadership. Again, lots of progress here. Our presentations, our posters at the MPS World Summit. We also present to regular animal users as well. We have a very broad reach. And we also have a one-pager on the FDA Modernization Act and MPS that we just recently released. The only other thing that I'll highlight from our organization is that we do have this technology hub. I'll post a link in the chat. It's super useful. You can see all of our different commercial members, sort them by tissue type, sort them by disease area, and quickly find a provider that works for you. For today, please do submit questions via the Q&A if you're able. If for some reason you can't access it, we will also be monitoring the chat. And then I also want to say that our next webinar will be on lung microphysiological systems on October 17th. We will be sending out invitations shortly for that, and I'll actually post a registration for that today in the chat. This is our tentative schedule. So after our representative from the IQ jumps on, we'll be having some consultants give us a really great overview of GI. Then each presenter gets about 15 minutes. They choose whether they leave some time for questions or not. But if you're able to in your question to actually direct it to a certain company or a certain individual, that's very helpful for us. And we'll have a round table with everybody at the very end. And with that, I will turn it over to my IQ colleague. Thank you.
Great, thank you so much, Megan. I'll go ahead and share.
Looks good.
Okay.
Go ahead.
Perfect. Okay, great. Hi, everyone. My name is David Kukla, and I am part of the IQMPS affiliate. And I specifically serve on our strategic partnerships and communications team. And I just wanted to start out by giving kind of a quick overview of the IQMPS. So the IQMPS is currently composed of 52 member companies, and we have representatives from drug safety, 3Rs, ADME, and PD/PK experts. And to the left, we have our 2023 leadership team. But besides the leadership team, we have a steering committee that is made up of 2 individuals from every company. And then to the right, you can kind of see a highlight of a little bit more about the IQMPS. We have over 115 active participants, which meet several times throughout the year. Additionally, we have 3 publications and are currently working on several more. Additionally, we created a new LinkedIn page, which is a great way to keep up with the IQMPS. And that continues to grow. Actually, I think the number listed here is a little out of date. I just checked, and I think we're closer to 350 followers right now. So the IQMPS has 4 main goals. One, to serve as a thought leader for both MPS developers and stakeholder organizations. Also, to provide a venue for appropriate cross-pharma collaboration and data sharing. Additionally, to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS in an industry setting, as well as develop external partnerships and collaborations to help enhance the inclusion of industry priorities. And the IQMPS is made up of 5 affiliate work streams, including manuscript publications, a regulatory outreach, a proof of concept studies through pilot project teams, a strategic partnerships and communications team, as well as a landscape gap survey team. And then here I just have an overview of our external partnerships over the past few years, starting back in 2018. However, besides that, we have several other activities within cross-farm collaboration, data sharing, regulatory agency engagement, as well as thought leadership. But some of the highlights from our external partnerships team, in 2021, we co-partnered with the NA3RC, which is now the 3Rs Collaborative. And with that, we established an MOU, which allowed us to work together on webinars such as this. Then in 2022, with the 3Rs Collaborative, we had 4 webinars on different MPS systems, such as neuro and liver. And then within this year, we also plan to have a total of 4 with blood-brain barrier and vascular already earlier this year. And then as Megan mentioned, we will be discussing lung in the upcoming month. Additionally, last year, we invited stakeholders to participate in an IQMPS FDA workshop. And during this, we discussed things such as when animal-based MPS may benefit the advancement of the field and the establishment of confidence in predictive nature of these platforms, as well as compare the perspectives of the IQMPS and FDA on animal MPS. And we also discussed providing examples where in vitro models are aiding in 3R's efforts. Overall, we're really excited to continue these efforts throughout the rest of this year, as well as the upcoming years to come. So if you have any additional questions on the IQMPS, please feel to reach out, or you could check out our website that I have displayed in the bottom left. But yeah, that's all I have for right now.
Awesome. Thank you, David. Hossein, if you want to come on with our first overview presentation.
Sure. Thanks, Megan. Yes, you should be seeing my presentation now. Okay,
All
great.
looks good.
Yeah, thanks. Such a delight to be here. Thank you, Megan. We are proud to be part of the 3Rs collaborative part of this webinar. My colleague Pelin Candarlioglu and I are going to give you an unbiased opinion about GI MPS, why and where you would need that, and at the end we are going to give you strategies, what kind of strategies there are out there about GI MPS. So first, we are going to give you a short introduction to us and who we are, and later on about GI MPS. So my name is Hossein Amirabadi. I'm a co-founder and managing director of Other Innovations, a company based in the Netherlands. We focus on education and training, specifically organs and chips, online and in-person trainings consisting of different modules. And we aim to help scientists adopt these technologies more efficiently, more confidently, and later on use these technologies efficiently. As well as connecting commercial MPS products to end users, again, more effectively. I would like to ask Pelin to continue the presentation about the GI MPS. Pelin, the floor is yours.
Thank you. Thank you very much, Hossein. If we can move to the next slide. So I would like to introduce myself a little bit for anyone we haven't met yet. So I am Pelin Candarlioglu, founder and director of 3D and 3Rs, which we are based in Cambridge, UK. So I would like to give you a little bit of a background what support and services we are providing at 3D and 3Rs, which is advice and management of adopting non-animal models, specifically organ on chip, to organizations basically anywhere in the world. And also, there's a wide range of organizations I can support, which of course ranges from startups, but also academic consortiums. For example, I'm a scientific advisor at the Smart OOC Consortium, and also a partner for a big Horizon grant that has been submitted yesterday, but also to large organizations, pharmaceutical or biotech. And the nature of the support can range from covering potentially technology development, but also its application, either in early discovery or late development stages of the pharmaceutical applications. So I personally have experience in having worked in multiple different types of organizations, having worked in also worked with, for example, global multinational corporations, either public listed or private, not for profit structures, but I have been worked very closely with SCRO Features or BBSRC, and also higher education and universities, both alone and also in the consortium networks. The areas of expertise that I offer, coming from my background, started with as a tissue engineer, and also worked both as an organ-on-chip developer myself and also as an end user. First track is value proposition of the technology. So this can be applicable to startups or technology developers by market positioning through defining their target group and scope of the application, quality and performance improvement of their platform in line with the expectations of the end users, and also creating a qualification criteria that is specific for the scientific question in hand, and also in line with the expectations of the end users. And also, quite importantly, model selection and development per application and scientific question, which could be quite attractive for smaller biotech companies. And another track of expertise that I can also offer as services is building and managing stakeholder networks. So this could be in the form of external engagement strategies, supporting product developments and roadmaps, and also monitoring competitive landscape, giving insights and aligning the product developments and business roadmap accordingly. So here is a very recent small selection of my professional and corporate experience. So in line with this, let's move into the GI. Can we move to the next slide, please, Hossein? Let's start and talk about why GI models are important. First of all, I would like to strongly stress about that using in vitro models for gastrointestinal applications in the pharmaceutical industry is not new. So in vitro gut models have been used in the drug development for many decades in ranging complexity for different workflows. But starting, of course, studying the diseases itself for efficacy applications, and on top of that, predicting drug absorption in PK/PD. Drug metabolism and drug-drug interactions are a huge part of the early in vitro models and why they have been developed. And of course, toxicity evaluation. So if we think about why we would need MPS in a whole gamut of these in vitro models that is already available. So there are a few niche applications where MPS provides a huge value because it provides a controlled environment in terms of cellular, biochemical, and biomechanical aspects. And we see in all these applications that I have just listed, the lack of these features have truly practical consequences in the therapeutics development. So I would like to expand this a little bit more. Can we move into the next slide, please, Hossein? Thank you. So let's consider an efficacy situation. We are studying the gastrointestinal diseases. So these days, there's a huge range of gastrointestinal diseases that is a big gap in the patient treatment and also in modeling. So, for example, inflammatory bowel disease is at the top for many pharmaceutical companies, and there's a huge need from the patient aspect on that. Similarly, colorectal cancer, multiple infectious diseases affecting the gut, and also the gut microbiome. It is on health management, but also from the pharmaceutical aspect, the drug absorption, drug-drug interaction, and drug metabolism is now known that these are aspects that could be affected and changed by the gut microbiome. And also predicting the drug absorption, which has direct implications at the PK/PD impact. So these are the immediate case scenarios where MPS can add value and does add value. On the safety aspect, immediately, specifically for the gut and gastrointestinal, toxicity evaluation has a huge impact. So I would like to invite you to read the beautiful paper that I have cited here from IQMPS, which is looking actually a specific context of use and how MPS can be used for looking into the side effects and mild toxicity of certain drug families in terms of whether they can cause nausea, vomiting, diarrhea, etc. And that paper is actually also excellent, how to create a qualification criteria for a specific context of use. On top of that, under the safety area, again, drug metabolism, which is closely linked with the drug-drug interactions, is one aspect where MPS can really add value, considering to what is available at the moment. And all of these that I have listed are instantly fall under the realm of the small molecules and fit perfectly. But also these days, especially for the last 10 years and almost exploding in the last 3-4 years, the realm of the biologics is opening a whole new venues. And the assessment of the safety, both for investigative and also at the clinical aspect, for biologics could be quite different than the small molecules. And this is also another aspect where MPS is really adding value. So now, having discussed the top high-level areas where we can see let's look into the specific case scenarios. For example, one of the main reasons where we have tried everything and that is not working and we need to go into the MPS is there's a clear need to mimic human physiology better. And that manifests itself more or less in the target expression. So especially for the disease biology under the efficacy subject, when target is not expressing at all on other preclinical model sets available, or it is expressing, but it is not expressing in a human physiologically levels, either in other complex in vitro models or in vivo, is a huge problem. And that exactly comes with that need to push up this target expression into human relevant levels. So when we see this problem, it usually leads into issues in twofold. One is, first of all, you cannot use a standard of care for that disease biology to benchmark your system. And secondly, on top of that, because you cannot create this benchmarking, then you cannot use your pipeline compound to test the efficacy. So these issues are twofold, and there has been examples and will be examples where MPS really adds value. And if we look at the safety, one of the most common case scenarios is there's a specific need to use controlled parametric studies in a human setting. Potentially because, for example, there's an unexpected response in the clinic and there's a clear mechanism of action studies is required to understand what is happening. So the problem in this situation is, first, the human data overrides all the preclinical, both in vitro and in vivo, and nothing that has been tried before, no other tools that has been tested preclinically is relevant to investigate this situation, because otherwise there would have been a signal already which would have been investigated. So this is a whole new situation that requires a new tool to investigate. So how we can do, what is available for us to try these different MPS systems? Can we move to the next slide, Hossein? So there are strategies to model the GI MPS, which Hossein is absolutely the right person to talk about. So I'll leave the floor to you, Hossein, to expand and enlighten all of us.
Great. Thank you very much, Pelin, for the very nice overview. So I'm going to be quick because the time is running out about these strategies. One point about the strategies is that these strategies are meant to really help you compare different models. So we have divided the design types to different types, and they are supposed to help you compare different models and think about advantages and disadvantages of these models from the practical point of view. I will quickly jump to the model. So the traditional model is basically 2D models in a Wells plate, epithelial cells. Usually, if you want to make it a bit more complex, you can pattern the surface. You can have them in 3D as organoids as well, and test compounds on them. But then if you want to create more mechanical cues, for example, create flow on top of the cells, you can integrate them in a simple channel to create physiological conditions for the cells. And that's the first type of the strategy that I'm introducing here. Now, here you can also integrate a lumen with different methods and have a lumen and create physiological conditions or physiological flow on the cells. Then one of the shortcomings of this model is that you have one lumen and studying transport is challenging. The traditional model is transwell here, but the transwell can also be microphysiological systems by integrating more complex biology, more cell types, more tissue architecture on different sides of a porous membrane on a transwell-like system. For example, here epithelial cells, endothelial cells, and you can study transport from the apical part to the basal side of this barrier model. Now, if you wish, you can integrate flow and mechanical stimuli on one side of the transwell or even two sides, and study also and add also circulating cells to the system, and study transport from one side of the membrane or one side of the barrier to the other side. The most important part is that there is a membrane, there's a porous membrane with cells on both sides of the membrane. Now, if you want to have more direct contact of the flow on the cells, you can introduce the membrane into a chip. So you have two channels on top and bottom of the membrane, and then you can also add circulating cells. And again, the transport, if you can study transport of molecules, but also transport of the cells through the barrier from one side to the other side, and the transport is vertical, but the barrier is horizontal. Now, the previous model was okay, but then if you want to have channels have the two lumens in one plane, so you could study transport in the horizontal direction, specifically for microscopy, then you can actually have different channels next to each other in one plane. But then most often you have a gel separating the two lumens from each other. The difference here is that the distance between the apical side and the basal side is often way larger than having a membrane. But then in the membrane scenario, you would have a vertical transport. The last case is basically connecting different organ models to each other. Gut could be gut liver, but it could also be other organs. And in this kind of chip, you would have, or in this kind of multi-organ chip, you would have different organ models connected to each other with channels. And in principle, you can use any of the previous strategies for the organ models that you're connecting to each other, two or more organ models. So this is an overview of different strategies. You're going to hear a lot. You're going to hear more about these strategies in the commercial presentations. And yeah, and this is an overview of different common strategies that exist. The floor is, thank you very much, Megan. The floor is yours. Megan.
Awesome. Thank you, Hossein and Pelin, for that great overview. If we want to have our representative from Mimetas jump on, and Pelin and Hossein, go ahead and mute. Reminder to all the attendees, you can always put questions in the chat as well as the Q&A throughout. Moran, I can see you. I can see your slides.
Yes. Can you hear me too?
I can hear you, and you are good to go.
Perfect. Thanks a lot, Megan. Yes, my name is Moran Morelli. I represent Mimetas today. And the technology that we use at Mimetas is called the organoplate. Yes, sorry about that. So the organoplate is a microfluidic platform. So it's a standard 384 well plate. We have different versions of it, but today I would like to focus on a version called the 3N64, which is the one we use for our gut-on-chip models. So as you can see on these pictures, it's a 384 well plate with a microfluidic layer underneath. And it's called 3N64 because it's composed of 64 chips in this version, and each chip is composed of 3 adjacent channels. That's what you see here on the right. So this is a close-up on one specific chip, and basically you can access the microfluidics through the wells of the plate. So through A1 and B1 you can access the left channel, and then through A2 you can access the middle channel in which we sit the extracellular matrix, and then you have another channel on the right which you can access through A3 and B3. The organoplate allows us to work with different setups. So you can seed cells in different ways with more or less complexity. So what you see on the very left is a single tubule. That's the simplest way. But then you can also have a tubule and some immune cells, for example, that are seated in the gel. You can have double tubules, or you can have a tubule with some cells around it. And today I would like to start with like the simplest version that we have with our gut model, which is the KCO2 tubule. So what you see here is how a chip looks. So you have a gut tubule on the top channel, then you have the ECM. So you can see that the cells are seated directly against the ECM because we have a phase guide technology which allows us to have a membrane-free system. And in the last channel, you just have medium. This plate then is placed on a rocker to generate flow, and what flow does is that it initiates differentiation of the KCO2, and we can get leak-tight tubule within 6 days, which is faster than what you get in static models like Transwells, because it's 21 days in those models. So once the cells are differentiated, they express different type of markers. So what you see here is an image of the tubule with like tight junction marker zona occludens 1 and the microvilli marker ezrin in green. But they also express transporters. And what you see on the right is a functional PGP assay in which you use the substrate of PGP. And then basically you measure in both the basal and apical side whether you can detect it or not. And what you see here on the left of the graph is that there's basolateral to apical PGP function, but if you go the other way around, you do not detect it. So basically it's pumping the right way. And then if you come with cyclosporin A to inhibit it, you see that you have no functional PGP. What the organoplate allows us to do as well is to simply image our chip. So what you see here on this picture is images of the bottom of the tubules. You have 64 tubules here, so each rectangle is one chip. And here I have two compounds. On the left, you have a compound that is not toxic. On the right, you have a compound that is toxic. The replicates are 4, so 4 chips per condition. So the bottom one is the vehicle, the top one is the maximal concentration. And in blue, you have the nuclei. In red, you also have the nuclei, but the nuclei of permeabilized cells, and you can see that the right compound is toxic, and is toxic in a dose-dependent way. Once you have 64 tubules, one application that you would like to sometimes test as well is the barrier integrity. And the reason for that is because most of the, like one of the hallmark of most intestinal disease is loss of barrier integrity. So to do that, we have what we call the organotier. So that's what you see on this picture. This here in there is the organoplate. So you really have a device that plugs onto the organoplate. So it's a device that is maybe this size. So it's really, really small, easily transportable from one hood to the other. And with this device, we can measure tear in every chip of the 364, and we can do that within 1 minute for the whole plate. And what is nice about it is that once it's plugged, you have no way of inducing variability as a user. So it's very fast and also very robust. One function of this organotier is also the time-lapse tear measurement. So what it does is basically to measure tear for example, every 1 hour for 24 hours. And that's what you see here on this graph. So on the x-axis, you have the time. On the y-axis, you have the tear. And it's just a starosporin dose response. So you can see that there's a dose and time-dependent response. I told you before that you have apical and basolateral access of your tubule through the different wells of the organoplate. And here, some of our customers in Austria, they basically use that to assess the response to a toxin. So it's a mycotoxin called deoxynivalenol. And their question here was, okay, does the route of exposure affect the response? So is there a difference whether like we expose from the basolateral side or the apical side? And you can see here, so on the left is the apical exposure, on the right is the basolateral exposure, and you can see that the response is different, and that in this specific case, basolateral exposure was more effective compared to apical one to decrease tear. So far, I spoke mostly about KCO2 tubule monoculture, but you can also easily add complexity in the organoplate. And here I have 3 examples. On the left, it's a paper that was published by us at Mimetas with tetraculture. And basically, it's an immunocompetent tetra culture where you have immune cells and intestinal cells that respond to cytokines. What you see in the middle is a paper that was published by our customers at Roche, who studied neutrophil infiltration, and on the right, still Roche, but this time with T-cell infiltration. So once you have this tubule, you can easily add complexity to the model. Another way of adding complexity is to get rid of the Caicos, because we all know that they have lots of advantages, but also some limitations. With patient-derived organoids, you can use a biopsy that is very specific to the region of the gut that you would like to study, and seed that in the organoplate to get tubules that this time are not Caco2 tubules, but patient-derived gut tubules. In this case, it's colon. And you can see that they also differentiate, and they form a polarized epithelium. So you can see here in red, it says ring, so it's a marker for microvilli. You can see that it's polarized with an apical and basolateral side. It also expresses tight junctions, MUC2 for goblet cells, which you don't have with Caicos, for example. Different microvilli markers, and lysozyme, which is a marker of Paneth cells. So what do we do with these models at Mimetas? Basically, we have three main applications. The first one and the top one that you see on that slide is drug development partnerships. So basically, we partner with usually a pharma company to sit down and develop together with them a model and its applications. Usually, those partnerships are long-term, so we're talking about years. The second one is a more classical fee-for-service, in which maybe a customer comes with a library of compounds and wants to test them in our system. So we do that for them at Mimetas in our lab. And finally, the last one that you see in the bottom is the customer that uses our technology in their laboratory. And to do that, we have what we call organo-ready systems. We have different ones with like vessels, or for gut, we have the Caco2, which basically is an organoplate that is ready to use. So you have the cells that are ready. We send them, you have 1 day of recovery, and then the customer can use them to challenge them with their compound and measure TIR, for example. I told you before that the 384 well plate is compatible with most microscopes. So what this allows us to do is to do some screening, and we started doing that recently. So here you see a robot pipetting in different organoplates. Unfortunately, we haven't done that in gut yet. I guess it's coming soon, but for now we have done it in vessels for angiogenesis, and there's a publication that was published recently in Angiogenesis, which basically goes over that story. So if we summarize gut-on-chip models in the organoplate, what you can have is up to 64 tubules without membrane, which are ready in 6 days, and you can easily access the apical and basolateral side. We also have readouts that are high throughput that can be combined, such as TIR microscopy, and also medium collection for analysis of specific proteins, for example. It's also very easy to lyse the cells and collect RNA, for example. And the modularity and easy access to the different channels allows to build complexity. So you can start simple and then add different cell types, for example. And yeah, with that, I'm done. So we already have some customers that use the organo-ready colon Caco2 as a gold standard, together with our organoTIR technology to measure barrier integrity, such as the SEIF in Davos and a group in Merck. And if you have any question, feel free to ask them now, or contact me or Silvano, our product manager. And if you want to have a look at the different publications that I mentioned, they're all listed here. Thanks a lot for your attention.
Awesome. Thank you so much, Moran. Yeah, so if anyone has any questions, feel free to put them in the Q&A and chat. Also, if somebody who's an attendee could just pop into the chat, just so that I can make sure that you guys can see it, even if you aren't able to put a question. Make sure that the tech is working today. Okay, thank you for that test message. The chat is indeed working. So if people do have questions, then yes, please put them in there. Seeing none at this time. I don't know if either of our consultants, whether Pelin or Hossein, happen to have a question for Moran. Otherwise, don't have to put you on the spot and we can switch to the next presenter. But you do have just a few minutes left, about 1 minute. So, okay, I will not put them on the spot.
Yeah, maybe I can ask a question, Pelin.
Okay.
If you have time, right? Yeah.
Yeah.
So I was wondering about the difference between the CACO2 inside the organoplate and basically statically. So what differences do you see there?
So the main difference we see is faster differentiation, which means that you can have your 64 chips and 64 lictal tubule that you can basically challenge after 6 days compared to 21 in transwells. And of course, in terms of experimental window, it makes your life much easier compared to static transwells. Also, the fact that it's just plugged in makes it very much easier to measure compared to static transwell, in which you have the chopstick. Each user does it differently. You can scratch the bottom. With the organotil, you don't have that. You just plug it in and you have no risk of damaging anything. It's non-invasive.
Awesome. Thank you so much. Awesome. So if you want to go ahead and turn off all your screens, and I will welcome the presenter from CN Bio.
Share my screen.
Perfect.
Perfect.
You're good to go.
Thanks, Megan. So the title of my talk today is Towards Improved Prediction of Human ADME Parameters, a gut liver MPS made up of entirely primary cells. So a bit different to what we just heard from our colleagues at Mimetas. So I'll be talking about our gut liver MPS and the integration of primary intestinal cells in our model. So why multi-organ and a gut liver axis? So multi-organ is really important because no two organs in the human body act in isolation. And that's particularly true for oral drugs that pass through the intestinal lumen as well as the metabolism subsequently in the liver. And as we go towards improved prediction of ADME parameters, we really want to better understand and model this in vitro. And also gut, liver and multi-organ systems are far better suited to mathematical modeling for better translation because you've got the different compartments that you're able to better define with volumes and with flow rates. And subsequently, gut liver axis is central to oral bioavailability, a really important parameter in ADME. When any new drug comes to market, you have to be able to predict oral bioavailability well, and this particular platform enables that prediction in vitro. But before I move on to the gut liver and integration of primary intestine, just a quick recap and introduction into our liver, because that's one component of the model. So for our liver model, we have primary human hepatocytes that are seated on a porous collagen-coated scaffold, whereby you have recirculated media going through the scaffold, through the cells, along the well and into the channel. And this is powered by our BioMimx platform, which powers the fluidics in our systems and in our models. So when we seed hepatocytes over time, they go from single cells, and as morphogenesis go from single cells into microtissues, and these cells are functional with expression of albumin, with staining of bioalkyniculi markers, as well as tight junction markers. And what's really nice is that these cells are functional over time in our model system. So if you look at CYP3A4 activity, our collaborators a few years ago, the FDA did a really nice paper whereby they demonstrated functionality of the primary hepatocytes and compared that to more traditional spheroids and sandwich culture. You can see here for both CYP3A4 and albumin, the expression is higher and you can express these functional markers for longer. So in terms of linking the liver and the gut for ADME studies, I'm going to introduce our first model that we developed, and that was on the hepatocytes, primary human hepatocytes, and the CACO2 HD29 cell lines. And the reason why we began with CACO2 model is that it's still dominated within the pharmaceutical sector, and it's it's quite sort of an easy introduction into this particular organ model before we get into primary cells. So in terms of this model and our dual organ plate, the way it works is that you can see the consumable plate on the top right-hand side here, and you've got 6 distinct wells. And on the bottom left-hand side, you've got a cross-section whereby you can see the gut apical compartment, where you've got intestinal tissue lining the membrane on a transwall. You've got the gut basolateral compartment, where you have recirculating flow, where media recirculates. The liver compartment, which is inspired by our liver plate from a slide I showed you previously, and you've got recirculating flow within here, and also a third pumping channel, which is our interconnecting pump, which transports media from our liver into the gut basolateral and then back again. And what was really important when developing this model was determining the functionality of both cell types within the dual organ plate. So on the left-hand side, you've got tier, and we looked at the tier of the intestinal model before and after the integration into the dual organ plate, as well as looking at albumin. And for both the gut and liver markers, both were maintained during co-culture in the gut liver MPS, which is a really nice starting point when you begin to develop these sorts of models in vitro. So one example that I've got with a drug, and it's always really nice to have a real world drug example, is the drug diclofenac. So for this particular example, we add the drug in the gut apical compartment. As you can see here, it permeates through and absorbs through the intestinal lining. There's then recirculation through the gut basolateral compartment, and then further recirculation as it gets transported in the liver compartment, where it then undergoes first pass metabolism and then subsequent transport back into the gut basolateral compartment and further recirculation. And within this experiment, we separated the dual organ plate into 3 different models. So our gut only, whereby you only have intestinal tissue in a transwell; liver only, whereby you've only got primary hepatocytes seeded and no gut model; and gut liver, whereby you have both the gut and the liver components together. And what's really nice is that when you've only got intestinal tissue, you get accumulation of the drug, but you see no subsequent metabolism. When you have only primary hepatocytes, you get a peak of the concentration of the drug much earlier, and that peak is higher. But when you have both the gut and the liver, you can see the combined effects of both intestinal absorption, but also subsequently hepatic metabolism. And what's nice is that you can then understand the different contributions of both clearance and the production of metabolites by intestinal tissues, hepatocytes, and then the combination of the two. In this particular example, you can see here the combined effects of both the gut and the liver. You saw more of the metabolite over each of the time points than we saw by both the liver and the gut when you add these two together. So that's quite a nice, neat proof of concept example. So from here, we then embarked into asking the question, well, how can we then improve the predictive potential of the model and integrate primary intestinal cells? So we began a bit of work to evaluate how we could do this, and we looked at various different options, from iPSC cells to taking primary intestinal epithelial cells and adding it to our model system. Last year, we began to talk to a company called Altis BioSystems, who have a talk later on in this webinar and workshop, and we began to talk about a collaboration. So we then entered a collaboration between our R&D team here at CN Bio, as well as the R&D team at Altis BioSystems. And they've got a really neat model whereby they've got crypt epithelium stem cells that are isolated from the human jejunum, which are then expanded on a biomimetic scaffold in static conditions. And on the left-hand side here is a cartoon of what their model looks like on a transwell layer. I'll leave the details to what their model entails to their talk later on. But as part of that evaluation and working with the RepliGut platform, we wanted to really understand the power of Altis's model. And we did a number of functionality tests, including histology, whereby we found that the RepliGut has a really nice, distinct and continuous layer of mucus, which you can see here with the staining of alcium blue compared to KK2 cells, whereby they don't have a goblet component and you get no mucus staining. So this is a really nice example of the functionality of primary cells, but also their cells when seeded on a transwell monolayer compared to cell lines like the KK2. So that we then embarked on working together and then trying to really integrate the both cell types together. And this wasn't a trivial exercise because we've got both primary cells from the liver and the gut with different requirements, different media requirements. We then managed to figure out, so the way we do it is by expanding the RepliGut cells and differentiate them in static to form a functional intestinal barrier. Once that's formed, we then add the transwells into the dual organ plate 4 days after hepatocyte seeding. And at that point, MPS and ADME studies can begin. And as part of that development work, we determined methods to ensure both cell types are functional. So on the hepatic side, primary hepatocytes are functional in co-culture with RepliGut. So you see here on the left-hand side, CYP3A4 and albumin, functional both in liver-only cultures and then in co-culture with the special media that was designed in the project. And then on the RepliGut side, the barrier is maintained in co-culture with hepatocytes, comparing both static and in co-culture. So finally, in this workshop, I'll just present to you a real-world drug example because it's really nice to see this in action. And the drug which we looked at was temocaprol. So temocaprol is a prodrug that gets converted into temocaprolat by the carboxyl esterase, which is a major metabolic enzyme responsible for the bioconversion of prodrugs. But what's really interesting about carboxyl esterase is their isoenzyme expression in humans versus cell lines such as KK2 cells. So there was a really nice paper back in 2005 which looked at both RNA and in protein. And what was very clear was the isoenzyme 1 of this carboxyl esterase is expressed in the liver, but not expressed in the small intestine and the colon. That's correct for both the RNA and the protein. However, KK2 cells wrongly express, whereby they express isoenzyme 1, whereas the small intestine and colon express the isoenzyme 2. And then when we were working with Altis, they shared some data to us looking at gene expression of their cells. And what was really encouraging, really nice, is that the jejunum matches the human isoenzyme expression. So you can see here in blue the expression of carboxyl esterase 2, whereas it doesn't really express the isoenzyme 1. So in practice, we added temocaprol. So on the bottom left-hand side here, adding it into the gut apical compartment or conducting liver-only experiments. What we can see here on the right-hand side is rapid metabolism of temocaprol by hepatocytes in our liver-only controls. Within 5 to 8 hours, most of the drug was metabolized by the primary human hepatocytes. On the bottom right-hand side, we can see the effect of transport across both the gut models in KK2 and in RepliGut was found to be quite slow from the concentration on the left-hand side. And that's in line with reported literature, KK2 permeability. And you can also see quite nice differences in the concentration profile when it comes to KK2 versus RepliGut. But the big question for us was, is there any clearance of temocaprol by the two different gut models? Do you see an effect of the different isoenzyme expression? And we did. So what we saw, and you can see on the top right-hand side figure, is the total molar fraction of temocaprol that was left in the entire system volume at 48 hours in all compartments, where we found that 99% of temocaprol was cleared in the KK2 model versus over 80% of temocaprol remaining in the gut-liver replicate model. And what you can determine from this is that most of the drug has been cleared by the KK2 cells before they were able to permeate through the barrier. And this isn't physiological for the KK2 cells because temocapryl as a prodrug is really designed to be resistant to intestinal hydrolysis. So this is a really nice demonstration, not only of the isoenzyme expression, but also an example whereby the KK2 cells completely metabolize the drug, whereas the replicate cells, in essence, are resistant to that metabolism of this particular drug. And finally, in the bottom right-hand side, we can look at temocaprylat, and for this, we looked at peak area. So this is the amount of the metabolite, or subsequently, as a prodrug, the parent drug that remains after 48 hours in co-culture. And you can see here most of it, it is the vast majority of it is then produced in the KK2 co-culture model versus in the replicate, whereby you don't really see much production of the metabolite, further confirming the correct isoenzyme expression in the replicate model versus in the KK2 model. So to summarize, the gut-liver MPS recapitulates the physiological conditions of oral drug dosing in the human. And one of the unique aspects of this particular model is that we take primary cells from both the liver and the intestine. And it's a really nice collaboration whereby we take Altis's unique intestinal cells and our capabilities of fluidics and multi-organ and expert in the liver, and we bring both of these together. And this sort of model is really useful, we hope, for the study of prodrugs and different ADME effects of oral drugs. So thank you very much.
So in the chat, I have just a simple question of what was the stain used to detect the mucus layer in the replicate?
So we used the Alcyon blue stain, which is quite a traditional stain used in histology for detecting mucus.
Okay, we're back on the schedule. Thank you, James.
Sorry about that. So anyway, Hesperos was founded by Mike Schuler and I about 8 years ago. We've licensed a bunch of patents from University of Central Florida, where I'm a professor, and Cornell, where he's a professor. We've used a lot of NIH grants to kind of really push the technology along. We've got a 14,000 square foot facility here in Orlando. We work with national, international pharma companies. We got about 50 people at the company right now. We're a CRO. We don't sell our platforms at all. But one key point is that we are the first microphysiological system where our efficacy data was the only data utilized to repurpose a drug for Sanofi into a phase 2 clinical trial. So we do have a lot of regulatory acceptance with these systems. And one of the reasons is we had this idea of clinically relevant functional readouts. And so really what that means is you go in a doctor's office, you immediately stick a needle in your arm, look at biomarkers, right? Looks for muscle contraction, electrical activity for neurons, cardiac cells, NMJ systems, barrier integrity, which I'm going to talk a lot about today. You know, you basically look, how's your elimination going and things like that. These are functional readouts. Allows us to non-invasively look at for acute, but more importantly, chronically. We've taken these systems up for 28 days. So what we can do, we can actually pattern and put neurons on top of microelectrodes, pattern cardiomyocytes over electrodes. We can put thin layers of cardiac cells or skeletal muscle on cantilevers, so when they contract, they deflect the laser. So we can actually then look at force. And again, we can still look at biomarkers, and we can also look at tear non-invasively in these systems. And this is how it kind of works. And this is going to be the theme of what I'm going to talk about. I'm going to talk about our gut models, but the real thing I'm trying to show is that we can integrate them into a multi-organ platform fairly straightforwardly. Because what we can do, we can actually take a base, we can take the different chips, depending upon what cell types, culture their own conditions, and then put them into a base. Then we put a gasket on top that defines the channels. And everything I'm showing you is all in serum-free media. We don't use serum at all. And we have a clear top, so if you want to do any kind of imaging analysis as well. And again, we can also do PK/PD modeling of the systems. We do CFD analysis to design systems, because
are we meant to only see your first slide? I'm starting to think that maybe, yeah,
No.
people are, okay. I just see your first slide. Wasn't sure if you were talking ahead, and then I realized maybe.
All right.
Yeah, we see like the PowerPoint not in presentation mode. I don't know if you somehow have it open twice. Okay, now I see. Yeah. Do you want to put it into
All right,
presentation?
I'm going to do it in non-presentation mode, if that's okay.
Fine too. Yes, absolutely.
All right, so this is the non-invasive functional readouts I was talking about. This is how basically the system works, where you have your base. We can put the various chips in. We can, again, if you want to take out a liver to see what the effect of metabolites with and without knowing what they are, we can do it. We can put in a gut model and take out one of the cardiac chips. Very straightforward to do. And again, we have a clear top, so you can actually then do imaging analysis as well. Okay. And again, we've worked with lots of barrier tissues. We worked with L'Oreal for years, so we can actually have skin models. We have kidney models in the system. We can use for tox targets. Mike could come up with one of the first blood-brain barrier systems with flow, and also he came up with one of the first GI tract models that had consistent activity over a period of time, and this is with CACO2 cells. We've now taken that model, okay, and we've created a BBB GI tract CNS 3-organ system where we want to look at transport, whether we do oral dosing, IV dosing, and then we have our control as intrathecal dosing. And this is what the system looks like here. Okay, and we have different channels depending upon whether we have the fluidic tract for the GI, the blood channel, or for the CNS channel. We can and then create the blood-brain barrier system against the brain microendothelial cells and astrocytes. We can then put these on inserts. So again, you see the GI insert for the staining for Claudin 5 and occludin and Z01, so we get very nice junction formation. And then at the same time, in the same media systems, we can have BBB, again, staining for the same tight junction markers. And the BBB, you can also see the glucose and the PGP transporters as well. Again, what we can do is we can maintain tier in these systems for both GI tract and BBB. And we can also show maintenance of the patterning of the neurons in the system as well. And be able to show we can still introduce long-term potentiation in the system and have that maintained, okay? So again, we can have a functioning 3-organ system where the GI tract is acting much more clinically relevant than an isolated system. And again, looking at the BBB, we can look at transport for PGP. We can look at bicuculin, which is normally transported very easily across the BBB. We can block it with verapamil until we get good functional activity, again, while maintaining tier in these systems. And again, different drug effects, we can have very stable tier over a period of time for both BBB and GI tract. And again, these are all monitored non-invasively for tier in the systems, okay? So obviously, we want to try to have an induced pluripotent stem cell derived model. This is all work by Mrudu Malik, who's our team lead for the barrier tissues, and basically showing that we have a modified gut model now, because we really want to look at the CYP activity in the GI tract, because people mostly look at metabolic activity with liver, but there's other tissues that also do a substantial amount of metabolism as well. And we have customers interested in what the percentage is for GI tract versus liver, and we now think we can do that. Okay, so again, showing the relevant biomarkers for the GI tract cells from the induced pluripotent stem cells. And again, just going in and looking at the same thing in terms of flow cytometry. So we get nice villain and mucin representation after about 37 days in culture. We can then check the viability of those systems. We can then compare it to a CACO2 model and show that our CYP activity is much, much higher than a CACO2. But then we can also do drug experiments, drug-drug experiments, where we can actually then inhibit that CYP activity with itraconazole, okay, and showing that we can actually look at drug-drug interactions in terms of inhibiting of CYP activity, because this becomes very, very important as well. And we can actually create dose response curves for that inhibition of the CYP activity in the GI tract, showing that not only we can look at barrier formation, but we can also look at metabolic activity of these systems in a multi-organ model. Okay, we can also maintain barrier integrity with the induced pluripotent stem cell derived systems. And also this is work that Dr. Malik had published, which he has brought to Hesperos, which is the incorporation of microbiome into the systems. So we have this capability as well. And this is being currently integrated with our systems. So again, getting to that whole idea of, where we can actually take a system. This is a heart liver system, but this could be very easily. We have Mike Schuler has published a GI tract liver system, but he really wanted to show you here is just looking at a heart only system. Okay, you see a large QT elongation for tetraphenidine, but its metabolite hexaphenidine does not have any QT elongation. You see it shifted when you put the liver in. Again, you look at the conduction velocity is not affected in the cardiac with its heart liver system, but heart only, you see an IC50 of 33 micromolar. And again, all this is done without affecting viability. We are really trying to look at functional deficits with our systems. We really try not to look at death markers except as an extreme endpoint. But the reason I'm showing that slide is that we can actually then take and model these systems. We can actually, this would normally be showing a model here showing the flow, but we can actually look at flow. We can look at shear stress in these systems because we can do the whole system in computational fluid dynamics. We can also look at absorption. Absorption is very, very important because all drugs will interact to some degree with all materials. You need to be able to account for that. So what we do, we actually then measure that absorption. For terfenadine, it's a fairly high. Hexaphenidine is fairly low. We can look at mixing, but we can then look at loss. But this is very important. If we're going to try to compare the results you're getting in the in vitro system to in vivo, you've got to be able to do area under the curve. Most people can only do as dosed. But what we do is we measure area under the curve. Where we can actually get PK/PD relationships where we can account for absorption. So again, absorption is not a bad thing as long as you understand it, can account for it in your model. And you can also look at metabolism. We can create the PK for that. Then in this particular case, we looked at the pharmacodynamics for the integration, the interaction with the outside of the cardiac cells. But the terfenadine interacts with the hERG channel from the inside of the cell. So you can actually get the pharmacodynamics from the inside of the cell in the system. And this data was published in Nature Scientific Reports in collaboration with AstraZeneca, where we used our in vitro PK/PD to predict in vivo animal PK/PD models for guinea pig, dog, and non-human primate. So we're taking this all the way from the in vitro to be able to try and predict the in vivo. And we've recently done that for malaria models where we can then predict first in dosing for anti-malarials in systems. And again, this is kind of what we want to try to do with these systems, is to take them all the way from basic concept, in vitro simple, in vitro complex, then predicting in vivo applications. Again, we can put them into multi-organ systems. We published this with L'Oreal in 2016 for drug interactions, 2019 for out to 28 days. We've also been able to put an immune component in for the innate immune system. This was published with Roche. But basically what we were able to do is to get circulating immune cells in the system where we were able to reproduce the effects for both restorative macrophages, or the M2, and the inflammatory macrophages, which was the M1. And we can get them to circulate in the system without being activated just by the circulation. Lastly, this is the folks at the company at our last Christmas party. I'd like to thank them. I'd like to thank you for your attention, and I'll take questions.
Awesome. Thank you, James. Thanks for your patience with all of the team's struggles. You do have one question in the chat. The person says, I might have missed this, but in the multi-organ setup, do you have a vascularized connection / endothelialized channel between organs?
No, we find it's not necessary to be able to do. What we're trying to do is to show the most extreme interaction of a drug with each organ, okay? And what we can do, we do have endothelial models, but they're separate compartment or a separate chip, okay? So if we want to understand the transport across the vascular system, we can then do that. And then we plug it into our in silico models. Because the problem is by having just the vascular system between the fluid stream and an organ, you leave out the lymphatic system, you leave out the interstitial space. Whereas what we can actually, if we can actually get that data for each one individually, okay, interacting with the same fluidic stream where everything is seeing the same amount, we can then apply, okay, the barrier systems in the in silico models to be able to predict the in vivo outcomes, which is really what we're trying to do.
Awesome. Thank you, Jane.
Of BioMimx. And today I will try to introduce you how we apply our technological solution to gut model. So to start with, in general, in BioMimx, our idea and vision is to provide a pharma company with new technologies that can help in discovering the therapies of tomorrow by starting from human data immediately. And to do so, we propose a technological solution which are beating organs on chip that, in our opinion, are able to unlock human preclinical model that are able to reflect clinical complexity. And of course, to describe you why we do it, let's start from understanding what in our side and in our opinion, clinical complexity means, because of course it can mean a lot of different things. But from our actually application, what we are focusing on is the fact that every tissue in the body is subject to motion. And you see here some examples. But not only this, also cells in human are also surrounded by a three-dimensional environment. And we believe these two components are essential to mimic most of the human organ in vitro. And for this reason, we developed a technological solution called beating organs on chip, which is able for the first time to couple actually motion with three-dimensional complexity by handling a unique in vitro setup, which is human, dynamic, and 3D. And you can see it in this picture. You can appreciate it, how the cell culture inside our beating organ on chip have a three-dimensional architecture, but are also subject to specific mechanical stimulation. And how does it work? You can appreciate from this video that sketch one of our functional units, in which from a cross-section you can imagine in blue our cells in 3D. And by actually moving a membrane, we can apply to these cells different kind of mechanical stimulation. Here, for example, are cardiac fibroblasts, which are stretched inside our model. And the beauty of our platform is also the fact that the system is extremely versatile, and we can have different configuration within the same setup. So if we take cross-section of a device, for example, you can imagine in the central channel to have cell in 3D that can be subject to stretching or compression by just changing the geometry of the solution. We can also have multi-channels, one close to the others, still subject to mechanical stimulation. And more relevant for this workshop, we can also use the setup in an inverted way and generate, in this case, barrier-like tissue that are subject again to mechanical stimulation. And all of this is now commercialized in a commercial setup called UBIT platform, which enable end user to start with the human cells, generate three-dimensional model inside the setup, subject them to the desired mechanical stimulation pattern, and then respond the effect to different drug, therapeutics, or pathological condition. And all of this is compatible with analysis based on imaging, for example, biomarker detection or gene expression, but also omics data. And in some models, and I will show you a perspective also on the gut, we can also integrate online electrical measurement in the system. But now, coming back to the core of our technology, why is mechanical stimulation important? Well, we show in a lot of different models that we can use mechanical stimulation in two ways. One is to use it to better differentiate and maturate human tissue in vitro by replicating mechanical drivers. And I will show you the gut as an example here. But we can also use it to induce and to mimic mechanically related pathologies. And we proved this concept in a lot of different publications. Actually, we focused at the beginning on solid organ on chip, so a lot of effort on cardiac and musculoskeletal model development and qualification. But lately, we are also applying the same technology to new R&D projects. And one of these actually that just came to the end is a study that we conducted on gut physiology in collaboration with Politecnico di Milano and the European Institute of Oncology. And actually, this work is all under review in Natural Biomedical Engineering, so I hope I will have the chance to share more data anytime soon. Diving into the application on gut model, the requirement we want to mimic inside our beating organ on chip in this case is the replication of the 3D architecture typical of the gut. So the William crypt structure and the multicellular structure encompassing both epithelial and endothelium compartment. Then, of course, and this is the key of our technology, we want to be able to show the advantage of adding mechanical stimulation, so replicating, in this case, peristalsis in the model. And the specific context of use we started to apply it for is the study of interaction of host microbiota in health and pathology. And I will show an example. So how do we integrate all of these components into our UGAT, which is our mechanically active 3D model of gut on chip? Here is the sketch, so you may recognize the layout of our UBIT platform, in which in a cross-section, in this case, we use the central three-dimensional channel to host an ECM gel, and we use the lateral channel to generate intestinal epithelium and endothelial barriers. Now, since the system is also compatible with perfusion, we can also perfuse bacteria or supernatant from bacterial on the intestinal epithelium compartment and immune cell in the endothelial compartment. And all of this is done into our commercially available platform and enables to replicate 3D architecture, direct interface of the two barriers, and to include mechanical actuation. And in particular, the mechanical actuation was developed by computational models that enable us to reproduce precisely the peristaltic movement inside our system, as you may appreciate from this video, with the hypothesis that peristaltic-like movement would favor the maturation of the epithelial monolayer, favoring the generation of villi-like structures. And to validate this, we started by seeding on the epithelial compartment CACO2 and HT29 cells, and we compare the generation of the monolayer in static condition or UBIT actuated condition. And actually what we could observe is that just after 5 days of mechanical actuation, we can have an epithelial barriers which present a three-dimensional like structure, which resemble villi-like configuration. And actually they express, they are high expression of Ki67, villin to show the generation of mature villi-like structure, and also mucin to they demonstrate also functionality and mucus production. And all of this is in a statistically significant higher way as compared to simple static 3D culture. Then, taking advantage of the modularity of the model, we also introduced the endothelial compartment. So in this case, we use human microvascular endothelial cells, and we generate a complete model encompassing epithelium and endothelial barriers. We characterize the functionality of the model, so we demonstrated that our two barriers are actually functional, and this by perfusing dextran at different molecular weight and showing that dextran is confined inside our two compartments. So as compared to a control model in which no cells are present, in our epithelial and endothelial barrier, the permeability is extremely low, showing actually the formation of functional barriers. Now, to apply this model to study inflammatory effect in the gut and eventually host microbiome interaction, we first saw that our UGAT model was able to respond to inflammation. And for this, we treated the epithelial compartment with LPS, which is basically a bacterial-derived endotoxin. And we could show that after applying LPS for 1 day in the model, the integrity of our epithelial barrier was compromised. We saw it by functional tests, so by using, again, fluorescent dextran, showing that actually is escaping from the epithelial compartment only when LPS is present in the model. But we also saw it from a morphological perspective, showing that the integrity of the epithelial barrier is compromised. And in particular, we saw fenestration in the epithelial barriers, reduction of ZO1 expression and E-cadherin expression. Now, taking advantage of the complexity of the model, so also of the endothelial compartment, we were also able to assess the effect of LPS on the endothelial side. And actually, we saw that, as expected, LPS treatment on the epithelial compartment had an effect of the endothelial cells, which were activated, as shown by ECAM1 staining, and also they were more prone to adhesion of immune cells. So we could perfuse T cell in the endothelial compartment and see how LPS promoted the recruitment of T cells in the model. Now, after having validated the model and showed that it was able to respond to inflammation, now we'll show an example in a specific context of use in which our collaborator at the European Institute of Oncology wanted to use the model to study the effect of microbiota-host interaction. And to do so, they choose to start with to study the effect of a bacteria, which is this Bacteroides fragilis, which is basically present in our gut in two different strains. One, which is not toxicogenic, that has been proposed also to promote intestinal homeostasis and actually as a probiotic factor. While the other strain is an enterotoxic strain, which produces an endotoxin correlated with intestinal inflammation and disruption of gap junction. So to see if we could recapitulate these two different effects, we expose now our UGAT model to supernatant collected from the culture of these two bacterial strains. And what we could show in a consistent way in the model is that when we apply supernatant from the positive strain, there is no any effect you can see on the integrity of the epithelial barriers, while the negative strain, the endotoxic strain or the endotoxin itself, drastically compromise the barrier functionality, showing that in this model we can specifically detect the effect of different host microbiome interaction. So to come to a conclusion, what I show you today is how in BioMimx we develop organ-on-chip models which are human, 3D, and dynamic, and are able to reproduce the complexity of the native mechanical microenvironment, like in this case, mimicking peristalsis. And how modulating mechanical signal enable us to induce maturation and physiological traits in a short time, like I show you for villi-like maturation. And specifically, I also show how our UGAT model could be applied for host microbiome interaction study. Now, as the next step, we are also applying the same model to different contexts of use, for example, to study drug absorption or the off-target, on-target effect of immune therapies on gut-specific tumors. And we're also integrating real-time tier analysis for a real-time assess of the model, and this by adapting a technology that we have widely validated in our cardiac setup. And just a final remark, what I show you today is offered by BioMimx in a service-based mode, and we also provide our UBIT platform under evaluation agreement in case you're interested to test and establish the gut model in-house. And with this, I would like to thank you, my team, and our collaborator in Politecnico di Milano and Istituto Europeo di Oncologia, and of course, thank you for your attention.
Awesome. Thank you so much, Paola. I have one quick question for you. In your static/actuated comparison graphs, do you mean by static, in parentheses, media flow/shear stress only, with no actuation/stretch forces, or static here means no stretch and no flow as well?
Yeah. So actually, this is the second point. So the static condition is with no stretch, but also no media perfusion. So we didn't test the perfusion of media inside the model in combination with stretching.
Awesome. Okay, thank you, Paola. There's one more question in the chat, but I'll let you answer that in the chat so that we can go ahead and move on to Chris. So.
Okay. Hi, everybody. My name is Chris Carman. I am Senior Director of Biological Development at Emulate. I think with this audience, I don't really need to introduce the limitations of existing sort of conventional intestinal models, and rather we'll just jump right into how at Emulate we're trying to deliver human relevant complex physiology in the intestinal model space. And since this is a 3 Rs webinar, we'll start with just a basic question. So what does it mean to develop a meaningful alternative to conventional intestinal models? So for us, that's clearly, first and foremost, developing and enabling researchers to capture human relevance with respect to cellular diversity, improved polarization, barrier function, and gene expression. Beyond that, we need to capture tissue specificity and recreate the tissue-specific characteristics and microenvironment, including mechanical inputs like flow and peristaltic stretch. And we need the model to have a broad ability to a wide range of use cases, such as testing drug efficacy, disease modeling, and ADME tox studies. And lastly, we need to capture flexibility and be able to readily tune the models to adapt them to the specific researcher's needs. So how can we apply organ chips to accomplish this? Let's jump into that. Here what we're looking at is Emulate's S1 chip, which features a 2-channel design, where our epithelial channel is highlighted in blue and the vascular channel in pink. And these are separated by a porous thin membrane. Here we're looking at a cross-section of our 2 region-specific intestinal models, the duodenum and the colon intestine. And what we're seeing in both of these is we develop parallel, analogous parallel workflows whereby human biopsy-derived colonoids are fragmented and seeded into the upper channel, and then tissue-matched either duodenal or colonic microvascular endothelial cells are seeded into the lower channel. These channels are each separately perfused with tissue-specific media, and then we can co-culture these cells in the presence of physiologically relevant fluid shear forces, as well as cyclic stretch, which is applied through these vacuum ports shown on the side. And this totality of co-culture and mechanical forces in this microenvironment helps to drive really strong differentiation and functional outputs for these models, as we'll step through in the next couple of slides. So let's start with the colon intestine chip model. Here, through immunofluorescence staining, we can see that we're able to capture all of the major epithelial cell types, including the absorptive enterocytes, gallbladder cells, and enteroendocrine cells. And moreover, using qPCR, we're able to quantify them and show that they're all within the expected ranges of quantitative percentages. And moreover, looking at a range of different donors, we're also able to show that we're capturing the expected donor-to-donor variability, which reflects what you would see in a real-world patient population. Moreover, if we look at multi-lineage differentiation markers as together with markers of stemness, we can see compared to traditional organoid modeling that we're able to improve the differentiation and maturation of these cells. Additionally, we're able to capture strong morphological maturation. As seen here, we're looking at the maturation of the brush border, which is indicated by a densely packed elongated microvilli. And in this micrograph shown at the bottom, you can see again that this cellular crosstalk, particularly involving the endothelium, is an important driver in capturing that fully matured phenotype. And the same is actually true when we look at development of the tight junctions and adherence junctions. The micrographs shown here capture cytoskeleton cell 1 adherence and E-cadherin. And we can see that these are really well formed and differentiated at the junctions. in the presence of endothelium, but when we take that away, they're much more diffuse and less well formed. And likewise, the functional barrier properties, which can be read out through a permeability assay, which the inverse of which reflects mature functional barrier, can develop over days. But when we see the black trace versus the blue, which is the absence and presence of endothelium respectively, we can see that the endothelium again is playing a critical role and helping to accelerate the maturation and differentiation. Now, cell height and polarity are separate but interrelated facets of epithelial cell functional maturation. And what this graph shows us is that on chip compared to transwell, and particularly in the presence of mechanical forces, including flow and stretch, are really critical for helping to establish this full maturation component. Now, in terms of gene expression, we can see through PCA analysis, again, that the full chip microenvironment with all the mechanical inputs really is critical in driving the transcriptome toward what you would see in vivo. So just look at the data points in red, full chip on day 8 compared to the teal, which is then vivo. You can see an extensive amount of overlap compared to the gray colloid transcriptome shown over here. So what's more is if we break that down and look at transcriptome in the absence and presence of endothelium, you can see that the endothelium is really critical for helping to drive that in vivo-like signature. So now let's turn to the duodenum. Similarly, by immunofluorescence, we can see that we capture all of the expected cell types. And moreover, again, compared to organoids in suspension, we can see that looking at specific differentiation-specific markers versus stem cell markers of stemness, we really are driving improvement in the differentiation in this full microenvironment. Likewise, we see that the right cells are there, but now we can look and see that they are polarizing in the way that we'd expect, and particularly looking at the sodium, potassium, ATPs, and the MDR markers, which reflect functionally important transporters and ion channels that are critical for duodenal function, as seen here. And likewise, we can see with the NHE3 and BCRP molecules are also showing the expected polarization, indicating that we're getting the correct functional output. Moreover, critical metabolic enzymes and receptors show improved expression compared to relative to organoid, and the same is true for critical efflux and uptake transporters. So what I've shown you some of so far is that the complex co-culture microenvironment with mechanical forces is able to really drive improvements at multiple levels of form, function, and transcriptome in these models. And as such, these models have really been widely adapted in many, many use cases in our customers, including more than 15 publications currently. And so what I'll do with the remaining time is just briefly walk you through 3 example use cases. So first is modeling inflammation. Specifically, we wanted to model the immune cell-driven pathogenesis reflected in inflammatory bowel disease. And to do that, we perfused inflammatory immune cells into the vascular channel at physiologically relevant shear forces, which are critical to drive selectivity. And we introduced IBD-specific pro-inflammatory priming stimuli. So what's shown on the left is a cross-sectional scheme of that process, and on the right is a cross-sectional confocal image of the resulting immune cell attachment to the vasculature and then migration to the epithelial channel on the top. Now, we're able to demonstrate that this recruitment was specific at several levels. First, it shows inflammation specificity. So if we perfuse the immune cells into a resting healthy chip, we get very little attachment or migration, as you would expect. But when we have the pro-inflammatory priming, we do drive an inflammation-specific attachment and recruitment of the immune cells that you would see in vivo. Moreover, we show specificity at the level of the tissue and cell types. So specifically in our blood and as we perfuse into the vascular channel of our model, you typically have 20 to 30% of the immune cells that are marked with alpha-4 beta-7 on their surface, which reflects a subset of immune cells that are specifically designed to serve the intestinal microenvironment. And what we see is when we extract the recruited cells that have moved to the migratory or to the upper channel, we see that those are largely enriched in alpha-4 beta-7 to greater than 60%. And that's precisely what you would see in a human biopsy of an IBD patient. So collectively, we're showing inflammation and tissue-specific recruitment here, which are exactly the sort of things you would like to see if you would if you want to develop meaningful IBD-specific therapeutics. And to that end, what we've done to validate that functionality is test a range of clinically relevant IBD therapeutics, including dexamethasone, AGM-300, Cimzia, and Humira, as well as not shown here, Entyvio. These are all clinically relevant and reflect a range of therapeutic targets and mechanisms of action. And I have time to just briefly tell you a little bit of the data we have for dexamethasone and AGM-300 shown here. And so if we focus on the recruitment onto the prime chips, what we can see is both dexamethasone and AGM-300 are able to suppress the recruitment. And what we can further see here is that at these saturating doses, we're able to rank order the efficacy of these two drugs. So now we'll turn to toxicity. And in this first case, we'll look at indomethacin toxicity. This is a well-known gastrointestinal toxicant. And on the left, we can see that we can demonstrate the predicted time and dose-dependent permeability response to indomethacin. And moreover, we're able to capture cell injury through assessment of LDH and IFAP-BP markers in the effluent. Lastly, the intestine chip was used to evaluate off-target, I should say on-target off-tumor safety risks of a candidate immunotherapy for colorectal cancer. And in short, in this model, by generating an immunocompetent scenario, we were able to capture the ability of the organ chips to accurately reflect the tissue-dependent target expression, as well as safety liabilities of a T-cell bispecific antibody in a dose and region-specific manner. So just to summarize here, I think I've tried to emphasize that by providing a complex microenvironment with mechanical stimuli and co-culture, we're able to drive human-relevant complex physiology. And I hope you enjoyed this brief dive into some of the example use cases that are emerging and supported by this technology. So with that, if there's any time, I would take questions.
Thank you, Chris. We are just at time for your presentation. I do see 2 questions in the chat, so I'll invite you to answer them in the chat. We can also bring them up verbally in the roundtable. And yeah, I'll invite Silvia online and invite Chris to go ahead and mute. And yeah, perfect. Go ahead, Silvia.
Thanks, Megan, for this. Thanks a lot. So we are React for Life. I'm the chief scientist and founder of this biotech company placed in Italy, in Europe, and we have patented and fully industrialized the organ-on-chip made MIVO. MIVO is the acronym of multi-in vitro organ device. We have used MIVO also for gut tissues. That is the topic of this amazing day. In the gut field, it's really important for our perspective to fully recapitulate the real complexity of this fascinating physiological niche in terms of three-dimensional complex tissues, multi-compartmental environment where we have the dynamic lumen environment and the bloodstream below, the possibility to properly control the flow with an high flexibility. And to do this, and in particular to overpass the limits of the animal and tests and the clinical trials, we have developed this organ on chips that is really highly flexible, as I will show later. And it appears with two different fluid dynamic condition named the single flow condition, so the flow mimicking the bloodstream below the gut tissue, and the second option is the double flow configuration with the flow even in the apical side, meaning the lumen side and the bloodstream. The most important feature of the MIVO device is its highly flexibility, even in terms of cellular models. And in particular, we have already tested and successfully validated the MIVO device in permeation studies by using different intestinal models having different level of complexity, starting from the acellularized membranes that are very useful for the early stage of pre-screening of molecules, then using a monolayer of cells, so immortalized cells, but also primary cells. We have currently an interesting collaboration with Altis, supplier of cells. And we have also already successfully validated the MIVO device, even with 3D reconstructed commercially available intestinal tissues, because we have understood that according to the need of the end users, it might be interesting to use different type of cellular models. And so using the high flexibility of the platform, we have validated different approaches as well. When we work with membranes, acellularized membranes, we typically use this configuration where you can easily host the membrane inside the chamber, generate a physiological flow condition below the membrane, and we have a nice sampling chamber where you can very easily and with a nice throughput sampling the media for drawing the kinetic of permeation on different molecules. Therefore, the benefits for this approach is the simple and faster readout, the high reproducibility, because typically the standard deviation of the kinetic of permeation is lower than 2%, and also the fully compatibility with a streamlined analysis. We have just submitted the papers where we tested the different molecules using this setup, fluidically connected directly with a spectrophotometric analysis to derive and obtain the kinetic of permeation since the beginning of the experiment. When it's important to test the permeation of molecules, not only through a passive route, but also through an active route mediated by cells, of course, it's better to use cellular models. And we have successfully demonstrated that when you introduce a fluid dynamic flow condition, the fully differentiation of CACO2 cells is faster, and it is completed at a shorter time and in very nice way way also in terms of tight junction protein expression. It's also important to stress that this configuration is fully compatible with the commercially available tier sensor, so it's also possible to measure the proper confluence of cells before starting the permeation studies. Interestingly, we have successfully validated another permeation studies by using a different cellular models. In this case, we cultured 3D reconstructed tissues and we tested the permeation of different molecules having different molecular weight and affected to different route, meaning paracellular and transcellular route. And we successfully obtained the proper ratio between lactulose and mannitose, whose ratio is an important clinical readout to determine if the patient has an healthy state or not. And if you see the comparison between the study condition of this intestinal tissue model and the dynamic condition, you may appreciate the very nice and better villi protein expression in dynamic condition and the very nice structure of the villi even under electronic microscopy. This was also a surprise by the supplier of these intestinal tissues. And the reason of this nice healthy state of the tissue is related to the presence of a physiological fluid flow condition feeding the three-dimensional tissue. Because if it's true that when you have monolayer of cells, meaning the red lines, either static or dynamic condition doesn't matter in terms of a proper diffusion of nutrients such as glucose or vice versa, the diffusion of molecules under testing. It's also true that when you move on a three-dimensional clinically relevant sites tissue models, so the blue lines, it's really important to introduce a dynamic flow condition in order to obtain the proper diffusion of the molecules into the three-dimensional tissue. And we successfully demonstrated what I said, both through computational fluid dynamics simulation and also through a chemical measurement of the molecules into the three tissues. When we culture the three-dimensional intestinal tissue, we also demonstrated that after an enzymatic treatment of the tissue in order to simulate the gut leakage scenario, it's possible to properly recover the healthy state of the tissue only in dynamic condition. Therefore, we obtain the first line of the absorption of the molecules, we damage the tissue, therefore the line was higher because the tissue was damaged, and then after the dynamic treatment of the tissue, the healthy state of the tissue was properly reestablished. This is extremely important because it opens the possibility to use this approach even to study in a more complex way the gut leakage scenario and the gut leakage assay. Even considering the fact that the MIVO device has a multi-compartmental device. So we have already co-cultured tissues like intestinal tissue, while immunocells were able to circulate under physiological flow condition. So we demonstrated that immunocells are able to circulate, to stay alive for many days, to not adhere to the walls of the tubes, and eventually, if they want, they are able to win the gravity force and the drag force due to the flow, and to pass through the barrier and enter into the tissue. Moreover, since this organ on chips allows to pick up the tissue at the end of the experiment, we also treated the tissues in order to collect the CD45 positive cells, meaning the immune cells compartment, and we further analyze these cells in terms of cytofluorimetric analysis and also immunofluorescence. Thanks to this approach, it was also possible to not only quantify the amount of the immunocells able to pass through the barrier and enter into the tissue, into the lumen, but also to enter and interplay with the cells colonizing the tissue. Importantly, despite other approaches, in this device, you can even culture many 100,000 of cells, meaning that you can even carry out omics and cytofluorimetric analysis for further investigation. Similarly, it's also possible to co-culture different types of cells, like gut and brain cells, in order to investigate even the interplay between gut and brain cells. And interestingly, since the organ-on-chips allow a separated sampling of the different compartments, meaning the lumen and the circulatory compartments, it's also possible to quantify the amount of the immunocells which infiltrated the tissue, but also the amount of inflammatory markers in the different compartments. And when we want also to introduce a bacteria into the environment in order to study also the interplay between microbiota and gut tissue, also in this sense, it's really interesting and important to have the possibility to sampling the media to different compartments, because in many diseases like inflammatory bowel disease, the final readout is indeed the passage of the bacteria from the lumen to the bloodstream, and vice versa, the passage of the immunocells from the bloodstream to the lumen. When we typically co-culture bacteria with tissues, we prefer to use the double flow configuration, as you can see also in this picture, because we have demonstrated that the double flow configuration allows to keep bacteria in a more predictive and in vivo-like environment. As you can see from the pictures, it's also possible to use different type of media, having different growth factors, pH, and media, and this is really important, especially for gut applications. We have the possibility to properly set up the flow, which is driven by a peristaltic pumping system. The velocity and the induced shear stress can be even different in the lumen versus bloodstream compartment, because we use different setup, and we can use different setup, and we can properly monitor the shear stress induced by the fluid flow in order to set up a physiological flow condition. And lastly, I want also to share the possibility with the MIVO device to generate multi-organ assays. So thanks to the high flexibility of the chamber, it's possible to fluidically connect multiple chambers, like in this way. So you can figure out to have intestinal tissue in one chamber and the liver tissue model in another chamber. Again, from the cellular perspective, there is a fully flexibility. So for the liver, you can use EBCI2 monolayer cells or liver organoids or patient-derived biopsies. And interestingly, you can study the crosstalk among different organs, but also the complete route and finally the oral bioavailability of the testing molecules, because you can sampling the media in the apical side of the intestinal tissue to determine the fraction of the intestinal availability after in terms of the solubility of the molecule. Then you can sampling the media in the bloodstream below the gut, and therefore analyze and quantify the fraction absorbed by the intestinal tissue. And after that, you can even quantify the fraction after the first metabolism in the liver. So it's really important to have the possibility to decouple all this fraction also for the molecule producer, because in some cases they need these parameters in order to optimize the molecule production. What I showed is what we have successfully validated in our laboratories, but we have already many customers among academia, pharma companies, and CRO that are already using this technology also in the gut application field. And so you can also have a look of their publication. And this is what we have done in the gut-on-chip application, but as a company, we have many other applications in many other different fields, and you can browse the website to know more. Thanks for the attention.
Awesome. Thank you, Silvia.
Hey, everyone. My name is Bryan McQueen, and I'm a senior scientist and manager of the commercial team here at Altis BioSystems. I'm going to talk to you about one of our model systems called Repligut Planar. It's a primary human intestinal epithelial model. Repligut Planar uses primary human epithelial cells isolated from the crypts of the human intestine. The harvested crypts contain both differentiated, which are non-proliferating, and undifferentiated, which include proliferating cells. The proliferating cells contain both stem cells and progenitor transient amplifying cells. So first, we expand all the cells isolated from the crypt, and through multiple passages, terminally differentiated cells and any resident immune cells are removed, giving us a clean and pure population of expanding epithelial cells. After several expansions, we can generate hundreds of thousands of vials of proliferating cells for storage. Then we can thaw the vials and plate them directly onto 12 and 96-well transwells, giving us the ability to perform high throughput testing. After a few days in culture, once the cells have become fully confluent, we induce a uniform switch across the culture, which promotes polarization and differentiation into different cell subtypes found in the gut epithelium, which I'll discuss a little bit more in detail in the next slide. But using this approach has several advantages. By controlling the progression from progenitor to differentiated cells across the culture, we have more control over the system to experiment exclusively on either expanding or differentiated cells. This allows for more mechanistic insight as opposed to systems where there are mixed populations of progenitors and differentiated cells, especially when these ratios are inconsistent across different wells. We also have complete access to the apical and basal compartments, as common with transwells, without having to use specialized equipment, which allows us to perform not only directional treatments, but also allows us to monitor barrier integrity through the culture life cycle using less invasive techniques. In our case, we use transepithelial electroresistance, or TEER. After we plate the expanding cells in our transwells, the cells are cultured for 3 to 6 days, at which time there's a culture-wide expansion of cells, as we can see here with EDU incorporation. This correlates to the cell population seen in the lower region of the crypts. And during this time, we can perform a range of assays exclusively on the proliferative cells. After the cells have reached confluence, we can change the media formulation to promote cellular polarization and differentiation. In less than 24 hours, the expanding population becomes undetectable, and differentiated cell subtypes such as enterocytes, goblet cells, and enteroendocrine cells become detectable. Additionally, we see increases in TEER and detect proteins associated with tight junctions, indicating tight barrier formation. Some of our quality control metrics are outlined here, such as time to confluence, the time to plateau start, as well as plateau length, which I'll also discuss a little bit more in detail in a couple slides. However, what you can see from this figure is there's a period of the maturation phase or differentiated phase in which the TEER plateaus. During this plateau is when we observe high number of the mature cell subtypes. As such, this marks the optimal window for experimentation on fully polarized and mature differentiated cells. I also mentioned that we've captured the polarized cells with histological sectioning as well. We do see slightly different kinetics in our small intestines epithelial cells with respects to TEER and cellular maturation from the colon epithelial cells. But for the purposes of time, I won't get into the differences today. To support Repligut Planner, Altis has established an ongoing intestinal epithelial biobank. All of our cells are isolated from healthy transplant-grade donor tissue. We isolate the proliferative cells from intestinal crypts from all region of the small intestine and the colon. All of our cells are frozen at low passages to prevent genetic and phenotypic drift that commonly causes altered cellular functions and responses, which we've previously determined by gene expression analysis, tier profiles, and response to inflammatory stem lag across different passages. Consequently, we freeze our cells down at very specific passages to ensure a long-lasting supply of cells for each donor without the concern for adaptation of cells to artificial culture. Demographics of our donor consist of male and female, Caucasian, African American, as well as different BMIs. These specifics are available with each study, giving our clients the ability to pick a specific region of interest and use the same donor and/or region in future experiments. All of our cell lots undergo very stringent quality metrics to become certified for use. We have metrics for vialing densities and freestall viability percentages. From the 4 available donors we currently have online, we consistently see viability percentages above 75%, with most being above 80. In this graph, each of these dots represents a generated cell lot from the respective donor. For growth kinetics, we've established metrics for time to confluence, which typically occurs between day 3 and 6 in culture. Any cell lots that don't reach confluence by day 6 in culture fail our QC and are not released for use. Lots that take longer than 6 days to reach confluence often begin developing these islands of spontaneous differentiation, which results in mixed population of expanding and differentiated cells. We also have specific metrics for once the cells switch over to the differentiation phase. We assess days to tier plateau, as well as plateau length. The purpose of these metrics is to ensure that each lot of cells we produce has consistent, predictable growth kinetics, ensures synchronized, expanding, and subsequently differentiated populations of cells across each culture. This also supports the ability to plan out experimental timelines. And of course, we test each donor for HIV and hepatitis, and all the cell lots are certified mycoplasma and fungi-free. More information about our quality control metrics will be released soon in a manuscript that we're finalizing now. Repligut has proven itself to be a very useful tool for preclinical drug development. In this collaboration with Yassen, we characterized the epithelial responses to a pro-inflammatory cytokine involved with IBD by measuring downstream chemokine expression and barrier integrity. As you can see, we were able to increase pathway-appropriate chemokine gene expression in our differentiated cells in response to interferon gamma. We were also able to prevent the increase in gene expression with the addition of the small molecule JAK inhibitor tofacitinib. In addition to chemokine expression, we also induced and prevented barrier compromise, as seen here with a loss in barrier in the presence of interferon gamma and prevention with the addition of tofacitinib, supporting the use of Repligut as a predictor of drug efficacy against immune-induced epithelial signaling and damage. It's also, Repligut's also a really great tool to evaluate cytotoxicity of drugs. In this collaboration with AstraZeneca, we analyzed barrier disruption caused by common chemotherapeutic drugs, OTX-15, topotecan, and afatinib. All 3 of these drugs commonly cause diarrhea in the clinic. And as you can see, each drug caused barrier disruption in a dose-dependent manner, supporting the use of Repligut as a predictor of epithelial drug toxicity, as well as a model to study therapies against these drug-induced toxicities. To take it one step further, we observed mechanistic specific effects of different chemotherapies. In this case, we determined the effects of bortezomib and colchicine. Bortezomib is a proteasome inhibitor that causes cell cytotoxicity in both the proliferative and differentiated cells in a non-selective manner. And as you can see here, this is exactly what we saw when we assessed total DAP-E counts in EDU cells. This graph right here shows a quantitative measurement of these observations, where the proliferative cells are the DAPI EDU positive cells, and then non-proliferative cells are those that are DAPI but EDU negative. In a dose-dependent manner, and before reaching the clinical Cmax, we saw a decline in both populations of cells. We also assessed the effect of bortezomib on proliferating and differentiated cell cultures independently and saw the same dose-dependent reduction in both populations. Colchicine is a microtubule assembly inhibitor, which means it really only targets proliferating cells by preventing progress through metaphase. Here we can see that colchicine decreased the presence of proliferating cells without affecting non-proliferating cells, which we can detect with positive DAPI staining and negative EDU incorporation. Quantitatively, we saw a reduction in the proliferating cells and no effect on the proliferating cells. When we applied colchicine to differentiated cells, we saw no effect on TIR. So together, these data sort of demonstrate the ability to not only observe toxicological effects, but also observe them in a mechanistic appropriate manner. As for the future of our cells and repligut planar, we have a lot in the pipeline, which I can't discuss at all in detail today. However, we're already partnering with different companies to supply our primary cells and help adapt them to different platforms with more applications. For instance, you already heard from our colleagues at CM Bio earlier about the great work they're doing by applying our primary cells and adapting them to their microfluidic gut-liver model. Additionally, we're always working on new models to recapitulate some of the complexities of the gut epithelium. Here is a representative image from our 2D crypt model, which we can grow 91 crypts in a two-dimensional format in one single transwell. As you can see, we can maintain zones of proliferation consistent with the cells of the lower regions of the crypts. And as we move away from these zones, the proliferative cells differentiate into subtypes consistent with the upper regions of the crypt, like goblet cells and absorptive enterocytes. We're also able to maintain an equilibrium in this culture of proliferating and differentiated cells for extended periods of time, which allows for extended treatments, washouts, and assessment of progression into specific cell types. We're also using repligut planar in its current format to create standardized offerings to be able to quickly screen compounds for different applications. For instance, we recently just launched repligut InflamaScreen to screen client compounds for potential therapeutic use against pro-inflammatory insults involved with IBD on differentiated epithelial cells. InflamaScreen utilizes cytokine dosings and endpoint assays that we have characterized, optimized, and streamlined for use with repligut planar. We can quickly assess the anti-inflammatory properties of different compounds by focusing on functional characteristics such as TIR and cytotoxicity. Additionally, we include a mechanistic readout with the offering, measuring pathway-appropriate epithelial chemokine release. This is important because what if you only assessed barrier integrity and that particular compound you're testing prevented disruption? While that would normally be good, if that compound enhances chemokine production, it could instead increase immune cell infiltration in the native environment, producing more pathological effects. So therefore, understanding the effects of not only epithelial cell health, but function allows for a much more comprehensive evaluation when screening. We designed InflamaScreen to accommodate up to 10 test compounds per plate while running it all in triplicate fashion, providing the statistics to ensure the results that we're seeing are genuine. For comparison, we have validated and include a clinically relevant FDA-approved therapy as a positive anti-inflammatory control against the cytokines as a reference for you to compare the anti-inflammatory effects of your compounds to. Because we've streamlined this offering and optimized all assay conditions and readouts, we're able to deliver the results in as little as 3 weeks from the time we receive your compounds. If you'd like to learn more about this offering or see example data sets and learn how it can be used for your screening purposes, please check out our recent webinar on thescientist.com titled A Novel Stem Cell Derived In Vitro Model of Intestinal Inflammation. Lastly, I'd like to highlight some of the downstream readouts we have optimized for Replica Planar. We have in-house expertise, equipment, and partnerships for a range of downstream assays, from some of the assays I've discussed already to things like drug transport, where we've recently partnered with another company for LC-MS analysis. We can also perform multiplex gene analysis using Fluidigm Biomark and Luminex QuantiGene technologies. We also can perform commercially available ELISAs, as well as multiplex cytokine analysis using Luminex. We also have the capabilities to perform up to 4-color ICC staining and perform high throughput and high content imaging. Any of these types of assays can also be customized to fit your specific project needs. Finally, I'd like to highlight that Replica is available in 12 and 96 well formats, giving our clients the ability to perform low and high throughput testing in relevant primary human cells. Replica Planar is also offered in kits, giving you the ability to use our systems in your lab. So thank you very much for joining and listening. Please feel free to reach out to us at info@altisbiosystems.com for any questions or project requests.
To start by briefly introducing Draper, we are a little bit different than most of the companies who've already spoken today. We are a large nonprofit, typically considered a government contractor. We do a lot of work for the U.S. government, but we are an R&D company. And one of the things I'd really like to highlight is the fact that we do have we are an engineering company, and because of that, our biology team, which is staffed with multiple biologists of many different backgrounds and trainings, but we work very closely hand in hand with bioengineers and various other flavors of engineers. And that gives us a real capability to create and build and iterate through different strategies to achieve the biology and bioengineering that we're trying to derive. And there's some details about us here, but I'll go on to introduce the device and platform that I'm going to talk about today. We do work with multiple different microphysiologic systems and also in a lot of different areas of biology. But in particular, today we wanted to introduce you to a platform that we call Predict 96. It is one of the main features of it that we'd like to highlight is that it's a high throughput system where we contain 96 individual platforms or individual devices, each of which houses a bilayer or 2-channel device. And all of these 96 replicates fit onto a standard well plate format. So in the top left, I'll introduce you to the structure of each of those 96 devices. There are 2 microscale flow channels separated by a porous membrane. And through each of these channels, we are able to flow. So we have independent flow through each of those channels. The entire system itself consists of that plate, which I've just highlighted, and then we have a lid, and that lid provides pumps that feed each of these individual channels. So we have 192 separate micropumps in the lid, and those feed through each of the channels, top and bottom, for the 96 separate devices. Some aspects of the Predict 96 plate that make it particularly useful for some of the readouts that we're interested in include the high optical clarity. So we're able to get really beautiful imaging done off of a confocal or off of even more high throughput automated imaging devices. All of the materials used in this are low sorption hard plastic. And so this allows us to work with materials without concern about absorption into the material itself. And let's see, on the pumping side, we can control the flow rate separately in top and bottom channels. And it is remote controlled, so you can put it into the incubator and set it and forget it, at least for a day, until you have to feed the cells. Because of the small format and the lack of spaghetti tubing coming out of the incubator, we are able to set up multiple of these platforms at the same time in a single incubator. The interface on the top of the plate does fit standard micropipetters, and so we're able to easily feed and sample within those chambers in a high throughput way. And we are able, as I mentioned, to do high content imaging and automated imaging in this format. So I'll introduce to you what we've done in this format with intestinal models. We have developed many different tissue type models, but in terms of the intestinal biology, we work with primary cells only. And as you've heard from a couple of other speakers today, we do derive them from donor tissue. We isolate stem cells, grow them as spheroids, expand them and can freeze them and biobank them. And we take those same spheroid cultures, dissociate and seed into the devices. We follow a sort of pattern similar to what you just heard about from Altis, where we expand the cells into the device for typically 4 days before we change media to differentiate them, at which point we see the tier increasing. In this case, I'm showing an example of our colon model where we can observe both formation of tight junctions and apical polarization through ZO1 and Ezrin respectively. We can see differentiation of MUC2 containing cells or goblet cells. At the bottom in the middle, you can see the increase in tier and the fact that we see relatively high tier in these colon models across out to 14 days post-seeding. So that's about 10 days after we differentiate them. And each of those different colored lines is a separate donor. You can see the consistency across multiple replicates of each donor marked by the error bars. We also have taken a look at the process of differentiation by evaluating the expression by RT-PCR of genes that typically mark stem cells, stem-like cells, so LGR5, ASCL2, and also an olfactomutin 4, and also Ki67 marking proliferation. So all of these decrease as we move the cells into a differentiation media and see those tight junctions forming. So one thing that we are interested in, we also have a model derived from ileum, from human ileum cells grown as spheroids. And we have characterized the fact that, in fact, the cells as grown in Predict 96 are able to retain the identity of the tissue from which they were derived. And this means that in the case of ileum, for instance, shown in the top left, we see, as marked by chromogranin A in green, we can see differentiation of enteroendocrine cells, which are typical of the ileum and not present in the colon. We did not see them in the colon. We see a lot more goblet cells marked by MUC2 in the colon, although of course we do see some in the ileum. And you can see some additional images of MUC2 staining and how we're able to get really beautiful close-up images of cells in these devices. On the right-hand side are additional RT-PCR measures of cell type markers. And this highlights the fact that we see when we compare colon to ileum, we are seeing markers of extensive enterocyte differentiation, which and so particularly the absorptive features of enterocytes, specific, much higher in the ileum. And also, as I already mentioned, the enteroendocrine cell markers. Paneth cells are typical of the ileum as well, and we see particularly those antimicrobial peptide markers, defensin A and Reg3 alpha, are particularly high in the ileum. And conversely, goblet cells, which are going to be a much higher percentage of the total cells in the colon, are again presented more high, are present at a higher level in our colon tissues than in our ileum. We also looked a little bit at differentiation of cell types in ileum versus colon. So just continuing that story where on the left-hand side, looking at additional mucin expression, MUC1, MUC4, MUC5AC are typical mucins for the intestine, and these are present at higher levels in the colon than in the ileum. And in particular, we have one case which is ileum B, and colon B, which are derived from the same donor. So these are true comparisons, and they are typically significantly higher in the colon than the ileum. We looked at functional capability of the ileum-derived tissue, and particularly we're interested in bile acid, the effects of bile acids on the cells. Bile acids are typically found in the ileum and resorbed there and not present in the colon. And indeed, we saw a sensitivity of the ileum cells to the increasing presence of bile acid as marked by increasing expression of FABP6, specific to the ileum, but not a response present in the colon-derived tissue. And also FGF19 similarly increased in response to bile acids in the ileum, not the colon. And then one story that I'd really like to highlight is some significant work that we've done around the colon and colon epithelium and its role as a barrier tissue and looking at barrier function. We studied this in two ways. We have tier meters that we built into these 96-well lids, and so we're able to measure tier across all 96 wells relatively efficiently. We've actually published that we're able to do that in a matter of sort of minutes to hours, so you can watch almost in real time the process of changes in tier. I think what this highlights is this is longer time periods where we've looked at the effects of TNF-alpha and interferon gamma. And because we have such high throughput, we're able to study the effects of these disruptors in dose curves against each other. And so we have two different donors across that 96-well plate. And in each of the 48 wells specific to each donor, we're able to do a cross-dose comparison of interferon gamma and TNF-alpha. And you can see the synergistic effects of those two features across as each one increases. And we've looked at two different time points, 12 hours after application, and then also looking at 72 hours, where you can see at the top we've removed the damage agents and see recovery in one of the donors, but don't see recovery in donor B nearly as well. We're able to take this density of data and actually calculate effectively a dose curve response, as you can see in the right. One thing that is really exciting to see is the reproducibility of this. So we also looked beyond tier, we looked at permeability, a dye permeability. So at the top, it shows basically a layout of how we run this experiment. We add dextran dyes, labeled dextran dyes to the apical channel and are able to then evaluate the transfer of this dye to the basal channel and sample out of that throughout the course of an experiment. We are recycling these, the pumps are circular, and so we can see consistent buildup over time and sample out of the channels through the course of an experiment. So we, in this case, added TNF-alpha to cause damage to the monolayers, and then looked at dye transfer through time. In this case, looking at a specific time and looking, we pulled a specific time to show you, and have 3 different sizes of dextrans that we looked at the transfer. And the interesting thing is you can see both the consistency among replicates, and you can also see how we're able to distinguish among the sensitivity of 3 different donors to the insult of TNF-alpha. Another aspect of the same experiment is that we were also able to evaluate the levels of IL-8 cytokine being secreted in response to the insult of TNF-alpha. So these are just ELISAs run on small amounts of media collected during the course of the experiment. And again, donor A, which was showing a lack of permeability induction in response to TNF-alpha, is also here not showing secretion of IL-8. So we're really able to characterize donor-specific sensitivities. So just in summary, I'll highlight that we have a high throughput capability here, which allows us to create a high density of data and really understand donor effects and look at wide dose curves of treatments. We do have this 2-channel device that we can accommodate multiple different cell types. In this case, we've focused just on the epithelial layer, but we have also started to integrate multiple different cell types. Again, I'll highlight that it's recirculation in each channel, and to date, we have worked with colon and ileum. We do have integrated sensors here I've highlighted today, but we're also introducing additional ones. And I think the reproducibility of the readouts that I've shown you is something that's very important to note. So thank you for listening.
Awesome. Thank you so much. If anyone has any questions for Elizabeth, go ahead and put them in the chat or the Q&A. And at this point, I will invite Janick to unmute share screen. My apologies for the previous issues. We can see your screen. If you just want to unmute and yes.
Perfect.
Okay, there we go. Thank you.
Thanks a lot, Megan. Very happy to be here. My name is Janick. I'm the co-CEO and technical director of Alveolix, and I'm happy to present you our gut-on-chip model, which is capable of recapitulating physiological 3D peristalsis. So we are a Swiss-based organ-on-chip company. We do have 2 locations, one in Bern, the beautiful capital, where we have our biolabs, where we perform internal research as well as service for pharmaceutical companies. And then we have our engineering and production site in Biel, which is also very beautiful, just at the lake, because everything is produced from us and then shipped to the world. So it's always clear that we need engineering and biology approaches to solve specific questions, and our engineering approach and our technology is shown here. So you probably know us from the lung field, but we also now further developed our lung-on-chip system to a so-called barrier-on-chip system, which is composed of 4 main components. We have the AX actuator, which is actually the module which is capable of recreating 3D stretching motion. We have the AX exchanger, which is another electron-pneumatic control system, which is capable of exchanging the cell culture media. Then we have the AX dock, which is the interface between the electric pneumatic control systems and our consumable, which is the AX12 inside. So the AX12 is based on the 96 well plate format, and you can recapitulate 12 independent organ barrier models in the system. And this is one unit. One unit is consisting of 3 wells. And if we look at the cross section, it looks like this. So we have an outlet well, we have a cell well, and we have an inlet well. Beneath the cell well, we have a very thin membrane integrated where we can create different organ barriers. Beneath, we have a fluidic chamber, which can be opened and closed through pneumatic valves. And we have our actuation chamber, which is then recreating the peristalsis in this case. So the membrane is made out of biocompatible silicone because we need to stretch it. We integrated 3 micron pores, enabling immune cells for migration. And we have a very thin barrier. It's only 3.5 microns, which allows us to generate ultra-thin co-culture models. And as mentioned before, we can also then add on different immune cells, for example, PBMCs, to make the biomodel a bit more complex. So how do we recreate the mechanical cues in our system? As mentioned, we have this gray actuation chamber where we can apply positive and negative pressure. This is allowing us to have the lower membrane in blue deflected up and down. And because we have a closed fluidic chamber, the upper membrane where we have the cells, which is then also reacting the same way. So here you see a movie of the 3D peristalsis, what we can recreate in our system. So this is actually up and down, really recreating the movement. The system can also be used for any kind of other barrier models where you are interested in studying mechanical stimulation. The application of the gut-on-chip is pretty clear. So if you have a healthy model, you can do safety testing. And then if you have a healthy model, you can also induce a disease state to do efficacy testing. And this is also what we've done with this model right now. So we started to actually create a model of a healthy patient. And here we used the standard cells, CACA2 cells. We looked at the static conditions in transvals and on our AX12. Looked at differentiation. We can also use tier because we have this open access from the top. We looked at gene expression, immunofluorescence to investigate the cell differentiation. So for the barrier properties, what you can see here, we have similar tier values in inserts and on the AX12, so meaning that we can have a barrier formation in our system. We looked at different epithelium markers for epithelium differentiation and also punnet-like cells differentiation. So we see that we have similar patterns of differentiation for the punnet-like cells. On day 14, we have a higher expression on the AX12, which is shown in green. Then we can do also high-resolution imaging. Here you see comparison of the cells cultured on the inserts compared to the AX12. So we see a bit more higher ABCB1 positive cells on the AX12, as well as a bit higher polarization of this marker. So we can create a robust barrier formation, and we can also increase specific markers, in this case at day 14, on the chip. Then the next step was to investigate the dynamics and also to further develop the monoculture system to have a co-culture system. So we used a monoculture system on the AX12 again in static, and we introduced HG29 also to have a bit more complexity in the model. And then we applied dynamics. And here in this case, what we did is we applied dynamics for 16 hours during daytime, so having the gut activity, and then 8 hours rest during nighttime. Again, we looked at the differentiation of the cells using tier, gene expression, and immunofluorescence, and we also looked at the enzymatic activity of the tissue. Here you see the gene expression of the cells which are not cultured on the chip, so just before. So what we see, we have enterocyte-like cells, we have pannet-like cells, and goblet-like cells if we combine the two cell types, CACA2 and HG29. And what you see here is the first effect of having the dynamics in. So what you see in the filled blue one, it's the static monoculture. In the open blue one, it's the dynamic monoculture. And what you can see is that including the peristalsis motion is bringing the tier values down. And that's also the case in the co-culture system, where we can bring the tier values down to more physiological ranges by having this stretching motion. Then if you look at the cell differentiation, again, in comparison with the cells which are not which weren't cultured before, so we have enterocyte-like cells differentiation in dynamic monoculture and co-culture. We have a bit less in the co-culture because of the HG29. We also have CDX2. Also there, we have a bit less in the co-culture on the AX12, but still we have a nice mix of these cells if we include also the pannet-like cells, which are highly expressed on the HG29, and we also see this expression then on the chip. We also looked at MUC2. They are highly expressed in HG29. We do also have a little expression then on the chip, showing that we have goblet-like cells, and we also did immunofluorescence where we stained the MUC2, which is also seen to be present. So we have with a co-culture, we have the possibility to have epithelium differentiation and have a diversity of epithelial cells. So this is why we decided for our healthy model, we're going to go with the co-culture. And then if you look also at epithelial functionality, the enzymatic activity is also more prone in the co-culture. Therefore, we then definitely decided that we are going for our healthy gut-on-chip model with the co-culture system to have cell diversity, and we still have functional barrier in tier and also having enzymatic activity there present. So the next step was going from the healthy model to a disease model, and here we induced an injury. So as mentioned before, we are doing CACA2 and HE29 now. We are looking at static and dynamics, and we are generating the injury, so the disease model, with a pro-inflammatory cytokine cocktail. Again, here you see the timeline. So we seeded the cells, differentiated them for 14 days, then induced the injury, and then 48 hours later, we looked at the readouts. What you see here on the left-hand side is changes in barrier properties, having the cells in static or dynamic cases. In the control, it's gray, and injury is blue. So what we see is we have a higher barrier decrease in dynamics. Looking at toxicity, we have similar patterns, static and dynamic. If we increase, if we induce the injury, the cytokine release measured with ELISA was more upregulated in the dynamic case compared to the static one, which is generating a larger testing window in the end. And we also confirmed with gene expression inflammatory responses. CACA-M5 is in both cases similar upregulated, whereas again here ILATE is highly more upregulated in the dynamic case. So we have this higher sensitivity to the injury in dynamic condition. So then we said, okay, the next step is to generate a benchmarking system where we use a drug which should prevent the barrier degradation via the injury model. And here, these are the culture parameters. So we only moved on for dynamics because we have a larger testing window, and we used azithromycin and 2 different concentrations. And again, the injury was done with pro-inflammatory cytokine cocktail. Here in this case, what we did is a prophylactic treatment using at day 4, integrating already the azithromycin, and then again at day 14, we did the injury, and then 48 hours later, the readouts. So what's actually happening with the barrier integrity? So here you see the fault changes. So we see a little decrease if you have the injury. If we use azithromycin at 70 micromolar, we have again more or less the same. But what we can see with azithromycin at 100 micromolar, we have still a very high barrier, so showing that the barrier is intact with this drug. If you look at cytotoxicity, so what you can see on left-hand side gray control, then the injury state. So we have an increase of LDH. With the treatment, we have a lower increase. We still do see a little increase of the injury acting on the cells. And what we see here nicely is, for example, the cytokine release. So we have a high release in the injury model, and then we have a dose-dependent decrease of the ILATE secretion using the different drugs. So with this, we have now a benchmarking system where we can test new compounds, and that's also what we are currently doing. So we're testing different compounds from a pharmaceutical company to investigate the barrier functionality increasement and also the decrease of certain cytokines which could be released. So in conclusion, we created the cell diversity on the chip with this co-culture, which is important to use and to model different biological variables in the gut. We integrate the mechanical cues, which increase the sensitivity of the system and the window testing. And we were also able to use qualified endpoints. So what does qualified means? It means that we can also correlate it to in vivo data. For example, barrier loss can happen due to diseases where you have diarrhea and fluid transport, and also inflammatory responses where we can also measure ILATE, for example, and different cytokines as well. So with this, I'm at the end of my presentation, and I would very much like to thank you for your attention, and I'm open for questions. Thank you very much.
Awesome. Thanks you, Janick. So that includes all of our presentations for today. Janick, if you want to stop sharing your slides. Great. And then at this point, we will enter into, either if we're going to have some questions. So our consultants will actually lead the roundtable. We technically have about 12 minutes left in the event officially, but we may extend if there are lots of burning questions. So I will turn it over to Hossein and Pelin.
Thanks, Megan. I think we have a lot of questions, right, Pelin? Maybe you can ask the first question, and I think it would be great if the speakers could turn on the camera and their microphone. Go on.
Yeah, absolutely. Thank you very much, Hossein. I do have a few burning questions indeed. I will just start with one. My first question is actually to Yassen from CN Bio. So it was extremely interesting to see what you have already accomplished with the cell lines. And it is always absolutely amazing to see further progress all the time. I was wondering whether you have considered the next stage at all, like including in terms of the gut-liver axis, including the portal vein, and to create a vascular barrier between the gut and liver, basically.
So I'm not seeing, so strangely, I'm not seeing Yassen online anymore. I don't know. I think though, yes. So I will actually pop on Darminder, who is from CN Bio. Hopefully you were paying attention to the question. If not, we can repeat it for you and you can come in as the rep from CN Bio. I know it's a long webinar, so yes, you should be able to unmute and share now, Daraminda.
Hello?
Yes.
Hi there. No, thanks for your question, Pelin. So I tell you what, I'll get Yassen to sort of answer that one, if that's all right. I think he just messaged and said he had to pop off, but I'll get him to answer and provide a response for that one.
Awesome. Thank you.
Perfect. Hossein, do you have any question? Maybe you take the next one.
Yeah, sure. Maybe for all the speakers, very nice presentations, by the way. Have you done studies, and I know you have, combining microbiome with the gut model? Yeah, I don't want to really start with the specific person, but if you have, please, yeah, give an answer. Thanks. Should I choose somebody? Oh, Paola.
I can maybe just quickly comment on this. So as I briefly saw in my presentation, this is actually the direction, the main direction in which we are going in BioMimx for the gut model. And we are taking different approaches for this. So the simplest one that I saw is to study supernatant, to insert supernatant derived from different strains of bacteria inside the model, and that's the first thing we did to validate. But actually, in collaboration with the European Institute of Oncology, we are studying the effect of microbiome taken from patients with colorectal cancer